So this is the review for Practical 2. The topics in this PowerPoint are going to be focused on the study guide questions. So if you want to go through it while you're answering the study guide or just checking your answers for the study guide, this is a good opportunity. So what is a culture and what is a pure culture? A culture is when you have medium, an agar plate, and it's inoculated with organism. And then a pure culture is the same, but it's only one species of an organism. So, for example, if you inoculated EMB with E. coli and there's no contamination, that would be a pure culture. The difference between agar and broth is that agar is the solid plates that we use in the lab, and then broth is liquid. So it would be in a test tube, and it usually has bacteria in it that we'd use to inoculate with. So types of culture media. You have selective, differential, all-purpose, and enrichment. Selective media will inhibit the growth of certain organisms while supporting the growth of others. So there's something in the media that causes other bacteria to not be able to grow at all. Differential, you're just looking at the visible traits that occur after your media has been incubated. So, for example, when you inoculate mannitol salt agar with Staphylococcus aureus versus with E. coli, there's a distinct change in the agar that makes it differential, and that makes it possible to differentiate between the two bacteria where they were inoculated. All-purpose media supports a wide range of microbial growth, so there's nutrients in it that support the growth of general bacteria. And then enrichment media boosts the growth of specific microbes. An example of enrichment media would be like the blood agar, which is used for growing fastidious bacteria, but the enrichment part is the blood. So, what type of agar is EMB? EMB is selective and differential. It's selective for gram-negative bacteria, so gram-positive bacteria can't grow on an EMB plate because of the methylene blue dye that's in the agar. And then it's differential for lactose fermentators. So E. coli, when E. coli ferments lactose due to the lactase enzyme, it produces a metallic color. The colonies have a metallic sheen, and that's how you differentiate the E. coli from other bacteria that don't have the ability to ferment lactose. And we'll get into cut that. Okay. So number 7 is, what are the ways to obtain a pure culture? You can get a pure culture from a streak plate, a pour plate, or a spread plate. And we do these methods to get isolated colonies. So when you do a streak plate, you're only getting isolated colonies. You can't quantify bacteria from that. When you do a pour plate or a spread plate, you use that for quantifying bacteria due to a number of colony forming units that you can calculate. The whole idea of the pour plate and the spread plate, you want to be able to calculate or estimate the amount of bacteria that was in the sample that you were using for the pour plate and the spread plate. So the difference between the pour plate and the spread plate is how you apply the bacteria. During the pour plate, you take from your dilution tube and you put it inside, you mix it with the agar, and then you pour it inside the petri dish so that colonies form within the agar. For the spread plate, you just take from the dilution test tube and you put a sample on top of the agar and spread it evenly. When colonies grow on the pour plate, they grow inside or within the agar in layers usually, and then when it's on the spread plate, it grows on top. And it should be isolated. They should always be isolated colonies for the spread plate and the pour plate. What is a serial dilution? A dilution of the stock solution. The stock solution is usually a container and it has a lot of bacteria in the solution. And so you'll have a series of multiple test tubes. And you're taking an amount from the stock solution, you're putting it in the test tubes. You're diluting it tenfold each time, usually tenfold each time. When you plate the dilution, when you plate from the test tube, that's another dilution. So you take 0.1 milliliters from the first test tube and you put it in the plate. That's a twofold dilution. The whole purpose of the serial dilution is to reduce the number of bacteria so you can get isolated colonies to form on the plate. The definition of a colony: a segregated clump. When you're looking at your plate, just whatever forms and it's round usually. The segregated formations are the colonies. When you're labeling your agar plate, you're going to label on the edges and you're going to label on the agar side to avoid, well, you go on the edges to avoid covering up the colonies that will form, and then you write on the agar side because when we incubate them, we flip them over, and you want to be able to see your label, right? And then, true or false, you should always blow on your inoculation loop. You should not do that. That's false. It says you should always blow on it after heating it. That would be bad because that would contaminate the loop with bacteria in the air from your throat, mouth. And then what is the purpose of a streak plate? So like you saw earlier, the streak plate is just you're swiping the bacteria and you're constantly reducing the numbers of bacteria that will form as you swipe it. So the whole point of the streak plate is to just get isolated colonies of whatever bacteria you're using. Tryptic soy agar is an all-purpose media. It's supportive of all bacteria, or most bacteria that you would inoculate on that kind of agar. Mannitol salt agar is selective and differential, and eosin methylene blue agar is also selective and differential. The MSA plates, they're selective because of the high salt concentration. So bacteria that can't tolerate a high salt concentration can't grow on them. And then they're differential for mannitol fermentation. The pH indicator is phenol red, and this changes to yellow in an acidic environment. So when you inoculate Staphylococcus aureus, which can grow on MSA, it ferments mannitol, which produces the acidic environment, which would change the pH indicator from red to yellow. I have here the results that you likely observed during the lab. So Staphylococcus aureus, it grows and changes the color, causes a color change. Staph epidermidis, it grows on the MSA plate, but it does not ferment mannitol. So the side with the epidermidis stays red. And then Pseudomonas aeruginosa and E. coli typically don't grow on the MSA plate because they can't handle the high salt concentration. So after talking about the mannitol salt agar, you're asked about the EMB plate, which I talked about in the previous slides. The main thing I didn't go over were the results. On the EMB plate, eosin methylene blue would grow. Pseudomonas aeruginosa would grow, but because it's gram-negative, but it wouldn't produce a green sheen. And then the remaining 2 gram-positive bacteria would not grow because of the because of the methylene blue dye. Okay. Biochemical testing. So biochemical tests are important for identifying a specific bacteria from a mixed culture and unknown culture sample. The Simmons citrate and the urea agar are both differential biochemical tests. So bacteria that can use citrate will produce a positive reaction in the Simmons citrate agar slant, and those that can use urea will have a positive reaction in the urea agar slant. The pH indicator for and the carbon source is citrate, by the way, for the Simmons citrate agar. So the pH indicator is bromothamo blue, and it works by, it's based on the change in the environment pH. So when citrate is utilized, the pH becomes, or actually the environment becomes more alkaline and basic, and this causes a color change of the pH indicator. The organism that's positive for the Simmons agar is Enterobacter, and the negative is E. coli. Urea is also a biochemical test and is differential. And it tests for the presence of the urease enzyme. The pH indicator is phenol red, and again, when urea is used, the conditions become more alkaline, changing the pH to be more basic. The pH indicator becomes pink. Proteus vulgarius is positive, so the slant becomes pink, and then E. coli is negative. The slant stays yellow. You want to know, so differentiation of Staphylococcus species. You're going to want to know the chemical reaction, or at least you should have an idea of the chemical reaction that occurs when catalase activity is present. So hydrogen peroxide gets turned into water and oxygen through a redox reaction. Okay. So a positive catalase reaction is the production of bubbles after adding hydrogen peroxide to the glass side with the bacteria. Staphylococcus is positive and Streptococcus is negative for catalase activity. During the staphylocide test and the coagulase test, you're differentiating between Staphylococcus aureus and Staphylococcus epidermidis. The staphylocide test is the detection of protein A on the surface of the bacteria. This works because there are antibodies bound to the latex beads of the slide. When the bacteria is added to the slide, the antigen, which is protein A, attaches to the antibody and causes agglutination, which is just clumping. If you remember from the lab, if you've gone over it at this point, the part with Staphylococcus aureus had blue clumps in it. The part with Staphylococcus epidermidis didn't have any. Similarly, the coagulase test, testing for a coagulase enzyme, which causes the plasma to clot. So in your test tube, you have a plasma sample, and when you add Staphylococcus aureus to the sample, it becomes thick. This is indicative of coagulase enzyme from Staphylococcus aureus. Staphylococcus epidermidis, there's no change in the fluidity of the sample. So number 2 of differentiation of Staphylococcus species asks about the 2 places where Staphylococcus aureus would normally be found. This can be found in your normal flora, like the nasal membranes or on the skin. And how do we differentiate between Staphylococcus aureus and Staphylococcus epidermidis? That was by using the coagulase test and the staphylocide test. So all the questions for the staphylocide test I went over in the previous slide. Especially E, it says which panel is likely to show Staphylococcus aureus. That's the one where there are clumps on the slide. The B sections, which asks about the type of test this is, I would confirm with your instructor what kind of test it is, because I would consider the staphylocide test a biochemical test because we're testing for a protein or an enzyme. I would also consider the coagulase test a biochemical test. You want to know that coagulase is the enzyme that causes plasma to clot, and Staphylococcus is positive for coagulase. So where do Streptococcus species normally live on the body? They can be found in the flora of the throat and the vaginal mucosa. So, which Streptococcus species are the greatest medical importance? That would be groups A through D, likely causing human disease. Streptococcus pyogenes can cause strep throat. Streptococcus pneumoniae can cause pneumonia. Streptococcus agalactiae is an issue for pregnant women because it can cause neonatal meningitis or septicemia of newborns. The Landsfield classification is our way of classifying different strep species based on the proteins that are present in the cell surface. So the study guide wants you to describe and give an example of different Streptococcus species between group A, B, and D. For group A, I would put Streptococcus pyogenes, which it is beta hemolytic and it's bacteriocin sensitive. Bacteriocin is just an antibiotic that the species is sensitive to. For group B, Streptococcus agalactiae is beta hemolytic also, and you can put the type of infections or diseases that they cause as well. Group D would be you'd be able to put Enterococcus faecalis, which is alpha, beta, or gamma hemolytic, which means it can either destroy red blood cells completely, partially, or not destroy it at all. And describe and give an example of bacteria that cause beta hemolysis. So Streptococcus pyogenes and Streptococcus agalactiae cause beta hemolysis, so you could choose one of those. Streptococcus pneumoniae is alpha hemolytic, and Enterococcus faecalis is gamma hemolytic, so you can choose that. So beta hemolysis is the complete destruction of blood cells. When you place a bacteria that's beta hemolytic on a blood agar, it will produce a clearing zone of the blood. Alpha hemolytic is only partially. So alpha hemolytic bacteria, they'll partially lyse the red blood cells. And for example, if you were to plate Streptococcus pneumoniae on blood agar, it would show up as a greenish brownish zone around the colonies instead of a clear zone. And then for gamma hemolysis, there should be no destruction of the red blood cells. So what type of media is blood agar? It's considered a differential media to observe hemolytic patterns of bacteria. So differentiation between Staphylococcus and Streptococcus, you want to be able to distinguish between their gram stain. They're both gram positive, so they appear purple after gram staining them. The difference between them would be that they're both coccus shaped, so they're both round, but when you're looking at them under the microscope, Staphylococcus would appear in clusters and Streptococcus would appear in chains. What rapid test can be used to differentiate between Staphylococcus and Streptococcus? That would be the catalase test, because Streptococcus is catalase negative. So superbugs and antibiotic resistance. A superbug is just a bacteria that's become resistant to antibiotics, and the resistance occurs through overuse or misuse of antibiotics by patients, anyone who has access to antibiotics. You're asked what is MRSA, VRE, ESBL. That's the antibiotic resistance bacteria here listed. So methicillin resistant Staphylococcus aureus, vancomycin resistant Enterococci, extended spectrum beta lactamase producing Enterobacteriaceae, penicillin resistant Streptococcus pneumoniae, and carbapenem resistant Enterobacteriaceae. I have a chart with their traits, their risks, and what they're resistant to. Even though it's described as methicillin resistant Staphylococcus aureus, they're not only resistant to methicillin. They're resistant to antibiotics that are similar to that, like amoxicillin. But these are how they're named. And then at the far right, you can see where they're found, how they can spread, what infections they can cause. Number 8 asks, is there one antibiotic resistant bacterium that is more severe? And that would be the Carbapinium resistant Enterobacteriaceae, because they're highly resistant and the antibiotics to treat them are very advanced. So they're hard to treat. Epidemiology is the study of a disease that's being observed in a population, and so epidemiologists study these outbreaks in these populations. You want to know what an index case is? An index case is the first case documented during an outbreak. Who are the two main think tanks of epidemiology? That would be the CDC and the WHO. Or WHO. What are the modes of transmission? So you have airborne, contact transmission, bloodborne transmission, and droplet transmission. The airborne transmission is through aerosols. So if someone were to walk inside a room where aerosols were, particles in the air, and they inhaled this or it went in their eye, they would be at a risk of infection. Contact transmission would be someone touching a fomite, right? So a surface that can cause contamination because there's bacteria on it, like a door handle or someone's pin. Bloodborne transmission is referring to pathogens that can cross over through the blood. So, for example, a nurse takes someone's blood and then the needle sticks her, she is at risk of getting a bloodborne pathogen. And then droplet transmission is like if someone were to cough or sneeze and it landed in your mouth or in your eyes and they're infected with some disease. And then which mode do you think is more responsible for spreading diseases? I'd say the contact transmission is just, it's easier for things to spread if they're not cleaned off of a door handle, a table, someone coughed and touched the table. And that's going to be the end of the Practical 2 review.